Hello, everyone. So, welcome to Pinoy Biotek Seminar, PCR Based Detection Kit for Salmonella in meat. So, we have almost or about 300 participants via Zoom and about 20 on Facebook Live. So, I am Lucille Punsalan from ISAAA Inc., and I will be the MC for this session. Thank you for joining us, and we hope that you will learn a lot today. So before we start, let me just share the house rules for this seminar. Okay, so we will first start with the activity objectives. So this seminar aims to discuss the challenges on food safety in the Philippines. It also aims to inform stakeholders on how the PCR-based Salmonella detection kit was developed and its applications and benefits in the food industry. This seminar also aims to learn about technology development and commercial pathways of similar technologies in the USA compared to the Philippines. So for the house rules, we highly encourage our audience to engage during the open forum, especially our online participants. So Zoom attendees are requested to keep their microphones on mute unless notified or requested. The online audience is requested to post their questions using the chat box later during the open forum. And please accomplish the online evaluation form to receive your certificate via email after the event. So if these are clear to everybody, we will now start our session with the opening remarks by Dr. Ian Fontanilla, the Director of UP Diliman Institute of Biology. Let's all welcome Dr. Fontanilla.
Thank you very much. And I'm quite happy and honored to co-host this very important event. This is a very important venue for our local scientists to showcase our technology, homegrown technologies. We do have the expertise, we do have the technologies right here in the Philippines, right here also in the university, and even here in the Institute of Biology. And two of our speakers are also affiliated with the Institute of Biology, and we're quite proud of them. And hopefully the third one will also be an affiliate faculty soon. And then I would also like to thank the participants. I know there are about 300 sa Zoom. Hindi para tayo magkakasya if we're going to do this strictly face-to-face. So I hope we will further engage with you and showcase with you the technologies that we are developing and how we can also inculcate or incorporate policies and for food security and food safety. So thank you very much also for, I have to say this right, the International Service for the Acquisition of Agri-Biotech Applications Incorporated, ISAAA, and also the Department of Agriculture, IB Biotek, also for allowing us to co-host this event with you. So again, welcome.
Thank you, Dr. Fontanilla. So next, I would like to call the Senior Associate Scientist of ISAAA Inc., Mr. Fanfilo de Guzman, to give a welcome message.
Thank you, Lucille. Good afternoon. Good morning, everyone. Apologies that our Executive Director, Dr. Rodora Romero Aldemita, cannot be with us today. She is currently in Canada and attending to some personal matters. So I'll read a message actually prepared for her. Pleasant good morning to all. Thank you very much for sharing your time to participate in this Pinoy Biotek Seminar. This is the third of the series where we provide a venue to showcase technologies developed by researchers supported by the DA Biotech program. Many of these technologies are in their mature stage and ready for release. And we hope that through this seminar, we will able to help sell the technology to target stakeholders and arouse the interest of the general public about the technology. Our featured technology for today is PCR Based Detection Kit for Salmonella infection in meat. This will be presented by Dr. Wendell Rivera, professor at the Institute of Biology. Before his presentation, we will first hear about the challenges in food safety, which will be discussed by Dr. Pirangeli Vital, head of the Biological Research and Services Laboratory, Natural Sciences Research Institute. And then Dr. Homer G. Pantua, co-founder and president of BioAssets Corporation, will share his expertise and experiences on technology development and commercialization pathways. Aside from the nutritional and health benefits from the food we take, food safety is also a concern. Safe and nutritious food is key to sustaining life and promoting good health. The increasing demand for food put greater responsibility on food producers, processors, handlers to ensure food safety. The detection of foodborne pathogens has been advanced significantly over the years with the use of different techniques and methods, both conventional, and advanced. Advanced methods include immunoassays, real-time, quantitative polymerase chain reaction or qPCR, for high throughput screening, biosensors, and the use of nanotechnology. The continued development of these detection methods for foodborne pathogens point to the importance of early detection and management of foodborne outbreaks, thereby enhancing public safety and health. Current and future directions should be focused on ensuring that existing tools are applied for foodborne pathogens control for use of food handlers, processors, and food surveillance and safety officers. More research should be carried out on the development of more effective approaches for the control and elimination of foodborne pathogens. Let me thank the Institute of Biology for co-hosting with us in this activity. We also appreciate very much our resource speakers for sharing their time and expertise. And of course, our participants, mostly our participants are online. And I hope we will have a very fruitful and engaging discussions. Thank you.
So now we will start with our first lecture, The Challenges in Food Safety by Dr. Vital. So Dr. Vital is currently a university researcher, scientist in the Scientific Career System of the Department of Science and Technology, Civil Service Commission, and head of the Biological Research and Services Laboratory at the Natural Sciences Research Institute in the University of the Philippines-Diliman. She is also an affiliate professor at the Graduate School of the University of the Philippines, Los Baños. She also served as an ASEAN Science and Technology Fellow for 2019-2020 under ASEAN Foundation and USAID, where she was selected out of more than 300 candidates all over Southeast Asia, and Senior Scientist Research Fellow in 2017, funded by the Department of Agriculture, Bureau of Agricultural Research. She is conferred as a diplomate of the Philippine Academy of Microbiology and recently received the following distinctions: 2023 National Research Council of the Philippines Gabay Award in Biological Sciences, 2023 UPD Galing Award, 2022 UPD Gawad Chancellor para sa natatanging REPS Research Category, 2022 Most Outstanding Kapampangan Science and Technology, 2021 National Academy of Science and Technology, NAST Outstanding Young Scientist in Biology, and 2020 Outstanding ASEAN Science Diplomats. Dr. Vital's significant accomplishments include her outstanding contribution to improving microbial food safety that promotes inclusive and sustainable development through her research work, publication, and the creation and adoption of several Philippine national standards. Dr. Vital represented the academic and research institutions in the technical working group of the DA Bureau of Agriculture and Fisheries Standards. Her expertise in microbiology resulted in the completion of several standards, the primary purpose of which is to provide science-based guidance on preventing, reducing, activating, and controlling foodborne hazards that present public health risks. Let's all welcome Dr. Vital.
All right, while waiting, I would like to thank the ISAAA and Dr. Adamita for inviting me to give a talk. And I also would like to thank academician Wendell Rivera for bringing me to this symposium for today. So my talk, I am assigned to discuss on the challenges in food safety. It is quite a broad or big topic. So I will be focusing specifically in the Philippines. I'm also glad to share or be sharing this topic because it is really one of my passion. So next slide, please. All right, so just to guide you with my presentation, this is the outline of my talk. It will be, of course, defining or a brief definition of food safety. And then the bulk of my talk will be basically, of course, on my assignment, which is on the challenges in food safety. And then a very brief discussion on sources and solution, and then what are the roles of the different institutions in food safety, such as the research, the academic institutions, and the government, and of course, takeaway messages or the summary. So at the end of the talk, as mentioned earlier by the different speakers earlier. So the goal is to, of course, share with you that food safety is indeed an important topic, not only in the country, but in all countries globally. All right, so first, of course, I go to the first part, which is defining food safety. And food safety is defined as the handling, next slide, please, preparing and storage of food products the best way possible to reduce the risk of contracting foodborne illnesses. So of course, it is a joint effort from the handler, preparer, and consumer to ensure the implementation of practices for food safety. So for those of you in the online, I think you can see that on the right side of the screen. So as you can see that based on the Centers for Disease Control and Prevention, the statement is everyone or everybody has a role to play in keeping food safe at every parts of the food chain. So that would be at production, distribution, processing, even of course during retail and the restaurant, and even in our homes. So there are different key facts in food safety. Next slide, please. And I would like you to take note that food safety is really important, not only as a food safety per se, but it is inextricably linked to nutrition and food security. So of course, nutritious food and safe food would be linked together. Of course, this would be affecting or impacting the economy and even, of course, our medical or health sectors. This is backed, of course, by the data from the World Health Organization that an estimated 600 million or almost 1 in 10 people in the world fall ill after eating contaminated food. And of course, children under 5 years, which are vulnerable population, would carry 40% of the foodborne disease burden or the daily. And then of course, on the right side, upper right, $10 billion is lost each year in productivity and medical expenses resulting from unsafe food in low and middle income countries. I say this not only in low and middle income, but even in developed countries, they understand that there is also cost and difficulty involved. Of course, needless to say that foodborne diseases impede socioeconomic development by straining healthcare systems and harming national economies, tourism, and trade. In a greater scheme of things, we can see, next slide please, that the driving forces that would shape future food systems. You can just actually see it as having the round at the center with the different factors affecting this food systems. I say it is demographic changes and the population movements that would be on the left. Widening inequality, the food sectors generates value-added growth and jobs. Of course, the fourth IR or the fifth IR would be rapid technological change and innovation. And then on the right side, evolving consumption, preference, nutrition, and health. And of course, the one that we realize now, it's climate change and resource scarcity. And at the bottom is localized development and strong stakeholder engagement, meaning that the cities and local actors are key to developing innovative solutions. So in a broader sense, food safety is not only food safety in itself, but of course it is linked to other factors which would change, of course, the food systems. It is just very easy to, next slide please, very easy to respond to food safety issues. The World Health Organization, years before, have 4 easy steps to keeping food safe. So just recently, they added the 5th step, and I am so glad that the 5th step is related to the field of microbiology. So the first 4 steps is keep clean, separate raw and cooked, cook thoroughly, keep food at safe temperatures, and use safe water and raw materials. As you can see, I showed 2 images because the World Health Organization has different languages for these 5 simple steps. So one of the languages is a Filipino, which you can see on the left side of the screen. So you can just download that from the World Health Organization website. Just how important the food safety is, not only for the country, but also globally. Next slide, please. We have what we call as the World Food Safety Day. So the World Food Safety Day is a yearly event that started in 2019 and celebrated every June 7. So the WHO uses this event to promote food safety to the public and reduce the number of illnesses that is caused by foodborne illnesses globally. In the Philippines, food safety is also important as we have the Republic Act 10611, or otherwise known as the Food Safety Act of 2013 of the Philippines. Simply, it is of course a long document, but of course the title is just an act to strengthen the food safety regulatory system in the country, to protect consumer health and facilitate market access of local foods and food products and for other purposes. And from this document or from this Republic Act, different agencies are involved in the IRRs, and as well as, of course, the development of the Philippine national standards for the different commodities and food products. And this would be under, of course, the Department of Health, particularly for the Food and Drug Administration and the Department of Agriculture. So I finished with my very quick introduction of what food safety is and how important food safety and how it is linked with other, of course, factors that would really impact the human health and the society in general. Of course, in the general scheme of things, it is part of the UN Sustainable Development Goals. So I move on to the bulk of my talk, which is the objective of the first talk, which is challenges in food safety. As of course, challenges in food safety is a bit general. So this is of course, minimized into looking at the agriculture and the aquaculture categories only. So I am referring to the aquaculture and the agricultural resources. Examples would be fish, shellfish, seaweeds, crops, and produce. I won't be giving much examples as I am not an agriculturist. So this would just be some of the examples that would be impacting our food safety. So what would be the story behind it? So for example, we were able to consume contaminated food. You could either have simple only, we could have food and waterborne diarrhea or other diseases related to eating contaminated food, or could be leading to outbreaks to pathogenic, can you please, thank you. Previous slide po. Thank you. Outbreaks due to pathogenic microorganisms. So it is essential to know that not all food and waterborne diarrhea or diseases are reported in our communities or even in hospitals. I know even siguro ako, I am guilty of not reporting if I feel diarrhea or any gastrointestinal disease. We do not report that we have eaten contaminated food. Although, of course, if outbreaks, there are already large reports of having contracted the disease. So that is only the time that we report that there is contamination of food. So this contamination is reported in different news reports worldwide, or I mean in the country. And these news reports are either written in the community or international or national newspapers, or even in articles that are written for publication. But then again, there are very low reports of other health diseases related to eating contaminated food. So I would like to categorize challenges into in food safety as having basically 5 factors or categories. And first one, I call it as a contaminant. So in microbiology, we define contaminant as something that makes a place or substance such as water, air, or food no longer suitable for use or for consumption. So in my case, if it's microbiology, this microbiological contamination referring to non-intended or accidental introduction of infectious materials or pathogens. I separate the pathogens because not all contaminants are pathogens. So the first challenge is contaminant, but the second one is pathogen. So as you can see, I categorized it as another challenge because it is an organism causing disease to its host, and the severity of the disease symptoms are referred to as virulence. It's comprised, of course, of different microbes, which I will be enumerating in a while, as well as different unicellular and multicellular eukaryotes. There are different sources of contaminations and pathogens in agriculture and aquaculture. And of course, as we know, in agriculture, we just categorize it as pre-harvest, during harvest, and post-harvest. This is in the belonging to the Department of Agriculture part. Now, if we process the food or if we cook the food, that is already belonging to the Department of Health part. So since I mentioned earlier that I am focusing on agriculture, so I focus only the contamination in pre, during, and the post-harvest. Sharing with you our results in the past researches and studies that we did, I emphasize water quality as having high microbial contamination in all types of crops or even in seafood products. Water quality would have really high microbial contamination. The next slide would be showing a bit of technical terms, but I still would like to define this because we cannot use it interchangeably when we are referring to contaminants, pathogens, and even how to attack food safety issues. So I just would like to differentiate a hazard and a risk. When we say a hazard, it is the agent, either micro, bio, or biological, chemical or physical, or condition of food having potential to cause adverse health effects, while the risk is a function of the probability of that adverse effect and the severity that the effect consequential to a hazard in food. So the one that I will be discussing would be the hazards. So next slide, please. What would be the commodities of concern? This would include, of course, the fish and other aquatic resources and the produce and the crops that I mentioned earlier. So why would they be contaminated? Because of course, for example, the fish and other aquatic resources, they are live organisms and become hosts and vectors to parasites and pathogens. And for example, we consume uncooked produce or vegetables. They can also harbor pathogens. And as mentioned earlier, they can be contaminated during pre, post, processing, distribution, and preparation, or even in food service or at home. And improper handling and storage would result to spoilage and contamination after harvest. Pathogen sources would include animal and human waste, as well as, of course, cross-contamination. The image would show how the contamination would go through. For example, there would be different animals. There are manure applications, the watershed, or even the irrigation system. So the potential pathogen transport of these animal waste, and even the waste from the manure, of course, would go to the streams or even in irrigation water. When we go to the sites or when we collect, for example, vegetables or crops, we can see that sometimes the irrigation water would not really be clean. They would just use, like, for example, available water, even sewage water to bathe or to give water to the crops. So that would really be a cause of contamination. So the common pathogens, as I mentioned earlier, I would be enumerating them. So this would be the big groups of the pathogens, which are the microbes, bacteria, protozoa, viruses, algae, and fungi. I will not be enumerating all of the pathogens. I won't be giving all of the examples, as I am only given a few minutes. So let me introduce just some of the food pathogens that we investigated and also of concern to us. So common food pathogen, first one is Escherichia coli. E. coli is one of the most common causative agent of foodborne outbreaks around the world. Of course, for those of you who are not biologists or microbiologists, on the right side of the screen is just a gram stain slide showing E. coli. E. coli, or most E. coli, of course, are not pathogenic, but some would cause severe illnesses. There are different pathogenic types, such as the famous one, which is 0157H7. I think it is the one that we hear in news reports, which would cause severe complications, such as bloody diarrhea, kidney failure, and even death. Again, for those of you who are not microbiologists, even in the online audience, the one on the right side of the screen are culture growth of E. coli, which you can see in MTech and in using methylene blue agar. So just briefly, or very briefly, a simple mode of transmission of E. coli, of course, this would be present in the lower intestine of warm-blooded organisms. We can either have fecal contamination of food or through undercooked meat. Then we can have gastrointestinal disease or a serious food poisoning. Another common pathogen is Salmonella. Next slide, please. I won't be discussing much of Salmonella, as it will be discussed by academician Windel. So Salmonella is, according to the World Health Organization, there would be around 550 million people affected with this organism, causing salmonellosis and enteric fever. Again, on the right side of the screen is just a microscopic view of Salmonella. A brief or a simple mode of transmission, usually in livestock, with either cross-contamination or food handling difficulties, so could have a disease such as salmonellosis. Another one is Vibrio species, which are abundant in aquatic environments, both in fresh and marine sources. So based on our previous study, these bacteria were all observed on the skin, gills, and the intestinal tracts of fish and shellfish. Again, on the right side is just a microscopic view. A very brief mode of transmission would be, of course, can contaminate fish and other fish products during improper handling, long-time transport, evisceration, and also cross-contamination from the raw materials, as you can see from the image on the right side of the screen. So I have already enumerated the first two food safety challenge factors, so contaminants, pathogens. The third one is foodborne intoxications or toxins. So foodborne intoxication is caused by the consumption of food contaminated with metabolites excreted by microorganisms. So a perfect example would usually be the red tide toxin. It is caused by a dinoflagellate, which is a microalgae that can be found in marine environments. So an example would be, it could be causing harmful algal bloom, and particularly they are red tides, referring to toxic blooms of microscopic algae that occur worldwide. So aside from marine harmful algal bloom, there are also freshwater harmful algal blooms that could also affect food safety, which is like cyanobacteria, giving rise, of course, to cyanotoxin. So for both marine and freshwater, pwede magkaroon ng harmful algal blooms. So toxins can target multiple organ systems, including the nervous system, the liver, the skin, and the respiratory tract. So you have contaminant, pathogen, foodborne intoxication, and the fourth one is antimicrobial resistance, or AMR. I call AMR, next slide please, as a silent pandemic. We don't see them, but the ARGs are being passed through from one organism to another environment to another organism, etc. So antibiotics have been used in different fields in agriculture, and it is used on plants and animals to combat diseases. However, of course, long-term use would promote AMR of bacteria in the environment. So, of course, as they are absorbed, they settle in excrement and are used as fertilizer in the soil. Okay, I just would like to show you very quickly the transmission dynamics, next slide please, of antibiotic resistance genes and bacteria among humans, livestock, environment, and produce, which was recently published by me and academician Windel. So this is the figure showing, as you can see, from the top, that is where the antibiotics is. There are different players, key players, of course, we as the organisms and the environment. The organisms, humans, even the animals, the environment, and of course the crops and the food pass through or transfer the different ARGs, as you can see from the diagram, the arrows that these antimicrobial genes are passed on and even interchanged that would go back and pass the antimicrobial resistance. So the fifth challenge in food safety is the chemicals. So I won't deal much with the chemicals as I am not a chemist also, but let me enumerate the chemicals that could affect food safety, as this is also, of course, a big challenge in food safety issue. So the first, of course, is naturally occurring toxins. You already mentioned foodborne intoxications, but other toxins would include, of course, mycotoxins and aflatoxins, which when we have long exposure could impact our immune system. We also have the POPs, or persistent organic pollutants, such as dioxins and polychlorinated biphenyls, or the PCBs, which could be found in animal industries. And this could, of course, be dangerous and would be affecting the immune system and would cause cancer in the long run. Of course, heavy metals such as lead, cadmium, mercury, present in toxic or in waste, having pollution, etc. And other chemical hazards. Examples would be radioactive nucleotides or even food allergens that are present in the entire food production system. So I have in total now the challenges as contaminant, pathogen, antimicrobial resistance, foodborne intoxications, and of course the chemicals. I would like to share with you another issue that we had in food safety, and this was food safety during the pandemic. We remember that during the pandemic, we just sometimes we cannot go to the groceries or we cannot go to the markets to procure our food. So we have meal kits and delivery. We have handling packaged food, handling and cleaning, and food donations. While there were no reports that COVID-19 were transferred due to these food deliveries and handling, but of course, other contaminants and pathogens were of course transferred, even the spread of the pathogens and the antimicrobial resistance. But we of course seen changes even in the packaging of our deliveries even up to this day. So who are the most vulnerable population to these foodborne illnesses? I say they would be the elders, children, pregnant women, and people with immune system. Although of course we are all vulnerable to these diseases caused by food safety factors or issues. In the general scheme of things, food safety, as I mentioned earlier, is not only food safety per se or in itself. It is linked with other factors of the society and the environment. As you can see, there's other factors linking food safety. And even with that, since society and environment is linked with the economy, so you can see if we will have or will be contracting diseases, it will be affecting the healthcare system. Of course, in developed countries or even in our countries, we have difficulty in the healthcare system. So with that, I have already answered the general objective of the talk, which is on the challenges in food safety. So let me discuss with you briefly the sources and solution of the food safety challenges that I have mentioned. So basically, since we are dealing with agriculture, I just be dealing with GAP, are the good agricultural and aquaculture practices. And these are measures to prevent or reduce the risk of hazards occurring during production, harvest, post-harvest handling, and ensuring animal and human health and welfare. So basically, I already mentioned the sources earlier, which are pre-harvest, post-harvest, and harvest or during harvest. So just to enumerate, these are the workers, animal waste, soil, water, animals, and sewage. And also, as mentioned earlier, during collection of several hundreds up to thousands of food products in agriculture, we have observed that microbial contamination is really highest in water, then in soil, then it decreases up to the crops and the produce. So just to see clearly, what would this be? What would these practices be to reduce microbial contamination? I really enumerated them because these are important points to see how we could answer the food safety challenges that I mentioned earlier. So first is on pre-harvest practice to reduce microbial contamination. Is to assess if the fish pond is free from, of course, the chemical or biological hazards that we have discussed earlier, to avoid entrance of farm animals, ensure that there is good water source, and of course, for example, you should have a sterile fry as starting materials, and so on also for the other food products. There are many pre-harvest practices. Of course, we want to reduce contamination at the source, at the point source. So more of pre-harvest practices, such as, of course, if there are outbreaks or if there are red tides, it should be reported immediately to local government units. And for example, if there's red tide or cyanotoxin tide, we notify the BFAR and avoid catching the shellfish. That is ideally. And of course, ensure, of course, human sewage is treated before releasing to aquatic sources. If water testing is available, tests should be done to identify possible contamination in water sources. And I think the last one is a bit hard to provide a safe alternative water source. As I have shared earlier, small farms or even mga urban gardens during the pandemic, I think our local government unit transferred or transitioned into having urban gardening. So even that, the irrigation water system was really very difficult to have it like having sterile water initially. So I think the last one is really very difficult. So aside, of course, so after pre-harvest, we have the harvest practices. So during harvest, equipment, of course, used should be washed before and after use. Harvest containers are checked and cleaned before used. After packing, containers should not be placed in direct contact with soil and water, and farm animals are banned where produce is handled, packed, and stored. As you can see, during the pre-harvest, the farm animals should not be there. But then again, when we go to the local sites or when we go to the farms, you can see that these animals are really roaming, not only the farm animals, but also yung mga alaga of the owners. Even the dogs, the cats are also there, and even the manure or their feces or the fecal matter could also be found there. So it's really very difficult to control contamination at this point. And then finally, we have the post-harvest practices to reduce microbial contamination. This would just be examples for post-harvest practices, such as packed container are not placed in direct contact with soil. Transport vehicles are checked before use, and produce are stored and transported separate from goods that may potentially cause contamination. Again, this is the Philippine setting because we are looking at the good agricultural practices and aquaculture practices, which are under, of course, the Department of Agriculture. And again, in the general scheme of things, ano ang sinasabi ng WHO? The World Health Organization response is to strengthen the national food control system to facilitate global prevention, detection, and response to public health threats associated with unsafe food. As you can see, I again enumerated them because these solutions are very important to target the challenge in food safety. So first is to assess the safety of new technology used in production, such as, of course, including genetic modification, cultivated food products, and nanotechnology. I think this was mentioned earlier. To help implement adequate infrastructure to manage food safety risk and respond to food safety emergencies through, of course, International Food Safety Authorities Network, and to promote food safety, including the 5 keys that I mentioned earlier. Next, to advocate for food safety as an important component of health security and integrating food safety into national policies and programs in line with international health regulation. To monitor regularly the global burden of foodborne and zoonotic diseases, at national, regional, and even international levels. And of course, update the WHO global strategy for food safety. It's not only the WHO working for food safety, but you can see there are other organizations. Yung pinaka-last line of the slide would be the Food and Agriculture Organization, the World Organization for Animal Health, UN Environment Program, and different US and UK institutions. And of course, we have the document Codex Alimentarius for food safety. So I move on to the next part, which is what are the roles of the different agencies or different institutions in the country to help in the food safety. So of course, I'm a bit biased, so I go first to the role of the research and academe in food safety. So I go straight to food safety in microbiology. But again, for those of you who are not microbiologists, microbiology does not only bring contaminants and pathogens or negative impacts. There are also, of course, positive benefits of course, beneficial microorganisms. The different techniques, such as the first technique, is a culture technique, which we use to study the contaminants and the pathogens that I shared earlier. And we also have molecular techniques. The molecular techniques are enumerated on the screen. There are so many molecular techniques now, and I believe our next speakers will be discussing these techniques. So all of these are being studied, of course, in the field of academia and research. And then what would be the role of government in food safety? It's very important that the government, of course, is involved in food safety, as not only, of course, the academia and research institutions, but of course, the government agencies would be the one first to implement the researches and even, of course, the rules, the guidelines, and the policies that we have. So effective regulations are needed to address food safety issues. And of course, this food safety, as I have mentioned earlier, is a shared responsibility of the government agencies. And in the Philippines, we have, when we talk of agriculture, it is the Department of Agriculture. I mentioned pre-harvest, harvest, and post-harvest stages. So example na lang, if you have kamote, harvest, binenta sa market, nasa market pa, dadalhin sa bahay, hindi pa niluto, that is under the Department of Agriculture. Now, yung kamote, naging kamote Q, niluto na siya, nagka-problema, dadalhin po siya sa DOH under the Food and Drug Administration. Of course, our local government units, such as the DILG, would, of course, help in the implementation of these guidelines, of course, at the local levels. And then, as far as I know, there are also food safety teams under the Department of Science and Technology. So I just would like to share with you my involvement in addressing food safety, particularly under a government agency. The Philippine National Standards is being done by the Department of Agriculture, Bureau of Agriculture, Fisheries Standards, or the DAPAFs. There are actually several, so many PNSs that you can download online, and of course the copies are with DAPAFs. And one of them, I am pleased to share with you that just late last year or early this year, we now have the primary and post-harvest food and feed product standard, particularly microbiological criteria. So all those pathogens that I mentioned earlier and all those different microorganisms also now has a set criteria or microbiological criteria when the food safety regulatory agencies and the different industries or even private and government industries would test. And with that, I go to my takeaway messages or summary. So I'd like to share with you the summary, my favorite summary from the World Health Organization. I say first, yeah, there are many opportunities for food contamination to take place, as we have discussed earlier. Contaminated food can cause long-term health problems, and food contamination also affects the economy and society as a whole. Next, some harmful bacteria are becoming resistant to drug treatments. We mentioned AMR earlier as a challenge in food safety. Consumers must be well informed on food safety practices. And the last one, which is really very important for us, which is everybody has a role to play in keeping food safe. So since my title is Challenges, there are new challenges to food safety that will continue to emerge. such as, for example, food safety fraud. And it is, again, another large topic that we could discuss aside from, of course, the food safety challenges that I mentioned earlier. And this is due because of our changes in food production and supply, including more imported foods, or even the global pandemic that affected our food safety. Changes in the environment leading to food contamination, new and emerging bacteria toxins in AMR, changes in consumer preferences and habits, and changes in the tests that diagnose foodborne illnesses. And with that, with everything that I said, I really just would like to leave you with this, that food safety really matters. It is everyone's responsibility, and safety is really linked to sustainability and food security, responding to the UN Sustainable Development Goals. All right, so I just would like to share with you our lab. Our lab is Biological Research and Services Lab. We do research, or one of the researches is on food safety. We also do service on food safety, such as cytotoxicity and genocytotoxicity. And thank you so much. Maraming salamat. I want to show my last slide because it is related to the. Thank you.
Thank you, Dr. Vital, for sharing about the challenges in food safety. Now we will proceed to the technology, which will be discussed by Dr. Rivera. Dr. Wendell Rivera earned his PhD degree in medical science, cellular and molecular microbiology from the Institute of Tropical Medicine, Nagasaki University School of Medicine, Japan. He is Professor 12 of the Institute of Biology, College of Science in UP Diliman. He was conferred the title UP Scientist 3 by the UP system in 2015, 2018, and 2021. He is recognized in the fields of molecular diagnostics and epidemiology, molecular protozoology, applied bacteriology, and medical biotechnology. He received various scientific awards in the Philippines and is ranked among the top 2% of the world's scientists based on composite citation indicators in 2020 and 2023. Dr. Rivera was elected as Academician of the National Academy of Science and Technology, Philippines, the highest advisory body in science and technology in the country in 2020. He currently serves as the chair of the Biological Sciences Division of NAST Philippines, chair of the Microbiology Consortium of the Philippines, president of the Phi Sigma Biological Sciences Honor Society, Philippines Incorporated, and co-editor of Sci-Eng-J, the official journal of the Philippine American Academy of Science and Engineering, or PAASE. Let's all give a round of applause to Dr. Rivera.
Okay, good morning, everyone. Uh, First, I would like to thank, of course, ISAAA for organizing this event, along with DA Biotech, the primary funding agency of the projects that we have in the past, probably a decade now, and also, of course, the Institute of Biology. So thank you. And as has been mentioned earlier, I'll be talking about the technology that we have developed in the lab. So we thought of this title to clearly show you a view of the things that I will discuss this morning. So it's technological advancements in Salmonella enterica studies in Philippine livestock, ensuring food security for future generations. So the earlier lecture given by Dr. Vital was a very good introduction, of course, to food safety, how we ensure food security for future generations. Okay. So just to give you an overview of the organism, again, for those who are non-microbiologists, would like to emphasize that Salmonella, the genus Salmonella actually has two species, one of which is Salmonella enterica, which everyone is very much familiar with. Okay, it's the one that causes human infections. And there's another species, probably you haven't heard of that, Salmonella bongori, but this one is quite common in cold-blooded animals. Okay, so it's known to be, Salmonella enterica is known to be an enteric pathogen. It's also zoonotic, meaning that it can, the infection can be transmitted from animals to humans. And as has been mentioned by earlier lecture, that is the cause primarily because of improper handling and also food storage. And we have to take note also that Salmonella enterica can cause gastrointestinal symptoms after incubation period of about 12 to 96 hours. So probably some of you were able to experience this. Okay, eventually it would cause diarrhea, vomiting, nausea, and of course, cramps. Okay, now other facts about Salmonella is that, next slide please. Sorry. Okay. It's estimated to cause approximately 93.8 million human infections and about 155,000 deaths annually worldwide. It can also cause major economic losses in poultry and also in livestock industries. And take note that serotyping or checking on the serovars are very important, especially for public health monitoring of the infection. The genus Salmonella is basically classified into serovars based on the presence of somatic or the O antigen and of course the flagellar H antigens. Now, interestingly, there's one technology that is commonly used worldwide. We call it White Kaufman-Lemonnier scheme, which actually gives us a view of the diversity of the serovars within the genus Salmonella, in particular Salmonella enterica. And the scheme contains an organized list of about 1,659 serovars. Now, you have to take note that livestock animals are really the natural reservoirs for Salmonella infection. So they harbor Salmonella, and this can, of course, be transmitted during evisceration, transport, and other production procedures. Okay, this was discussed earlier by Dr. Vital, and clearly we can see here that in the food production chain, Salmonella enterica, being an enteric pathogen, can be transmitted, of course, from animals to humans via this chain. So you're familiar with this. Of course, you start with the farm animals, okay, and then it goes through the abattoirs or the slaughterhouses. They are processed. And then, of course, after processing, they are transported to different places, okay, in supermarkets, for example, until they reach the consumer. Okay, so the infection, contamination would happen anywhere during that production chain. Okay, so it's basically contamination can come from pre-harvest, as mentioned earlier, during the processing of the food product until post-harvest. Okay, so now let's talk about the methods for Salmonella detection. So this is quite common, especially here in the Philippines. Of course, there are monitoring agencies that really check on Salmonella contamination. Okay, prime of which is, of course, the National Meat Inspection Service, or the NMIS, which is the main monitoring agency for meat products, okay, being sold here in the Philippines. So normally they would use this method that would take up to, okay, if you would like to complete, it's up to 7 days, of course, to check on the serovar of a particular organism. But normally it would take probably 5 to 6 days. So normally it starts with enrichment, because you need to multiply and check for the detection limit for your salmonella. So you need to enrich it. And normally when you enrich, you take a small sample of your meat product, say 25 grams, and then mix it with a buffer solution, like a buffered peptone water, to basically pre-enrich, okay, and allow the organism to multiply that enrichment medium. And then the next step would be selective enrichment. Basically uses this time a selective and at the same time a differential medium, one of which is Rappaport Vasiliadis broth. And it's selective, and at the same time it differentiates the salmonella with other gram negatives. Okay, and in some cases, of course, after RV or the Rappaport Vasiliadis broth, they are a portion of which can actually be inoculated into another medium like XLD or the xylose lysine deoxycholate agar. And this time it also differentiates a salmonella from other gram negatives. So there's a need to plate, of course, and there are obviously characterization procedures that need to be done, and also some biochemical tests to make sure that the isolate is really salmonella. And if you would like to go to the serovar identification, then you have to go through that last step of serological test. Okay, so take note that this is the, until now, this is the gold standard of salmonella detection, and it takes up to 7 days to basically complete the procedure. Okay. But then, of course, there are advantages and disadvantages, but we would like to highlight, of course, some problems with the current procedure that we have. Okay, so we are using basically culture-based and biochemical methods, and we know that while culture-based and biochemical methods remain important tools to determine the microbial load and the presence and absence of foodborne pathogens, these also have several disadvantages. And we have mentioned here 3, one of which is the long turnaround time. This is quite disadvantageous because of the products that need to be processed. Okay, they are highly perishable goods such as meat. So as much as possible, we have to be really fast in identifying the contaminant, okay, the microbial contaminant in particular. Also, of course, the procedure is labor intensive. It involves a lot of culture media preparations, okay, plating, and the procedure is really tedious. So this is another disadvantage. And lastly, results are limited to the presence or absence. Okay, you cannot go deeper into checking on the virulence or even pathogenicity of the organism, and also antimicrobial resistance. So if you are into those information, then of course you cannot use merely culture-based and biochemical methods for that. So due to these limitations, faster, more cost-effective, and more efficient detection and characterization methods are, of course, needed. So with this, we had this publication in 2020. It's on the PCR-based detection and serovar identification of salmonella in retail meat collected from wet markets in Metro Manila here in the Philippines. And in this publication, we were able to describe a much faster way of detecting the pathogen Salmonella enterica from meat samples, okay, from retail meats that we collected from different wet markets here in the Philippines. And it's quite faster than the traditional method of doing it in 7 days. So how does that procedure work? Okay, so from the 7 days, we were able to do it much faster in 3 days. Okay, we still have the pre-enrichment or enrichment protocol, okay, that would use the buffered peptone water or BPW again to multiply the organisms present in the meat product. Okay, and then we go directly to selective enrichment, and that would basically use the medium Rappaport Basiliadis broth. Okay, and then after which, okay, since it's a broth culture, we take microliter amounts, okay, from that broth medium, and then use that amount to basically extract your DNA. Okay, so your process, the usual DNA extraction would be to you to centrifuge and let the pellet be used for further extraction. And it's quite easy to extract DNA, especially in bacteria, gram-negative in particular, because you can use the usual boil lysis method. Okay, so you lyse by merely subjecting your bacterial pellets in a boiling temperature, 100 degrees, and voila, you have, of course, your DNA afterwards. Okay, and then you extract after extracting your DNA, you subject it to the usual polymerase chain reaction. Now, what we're doing is to basically use a gene that is unique to Salmonella enterica, and we call that the INVA gene. It's a, a small fragment of a DNA in the whole genome. If you do PCR, your amplicon size is just around 244 base pairs. So it can easily be detected by agarose gel electrophoresis. So what we did was to, of course, establish this method, and at the same time, validated it to be proficient, okay, in the following matrices. We've checked beef, chicken, animal feeds, also cotton swabs. But of course, there's a challenge here. More matrices must be added to this roster for this method to be fully adopted by testing laboratories nationwide. Now, when we say we validated the procedure as proficient, we mean that we actually used a third-party company, okay, and later on I have a slide on that. It's the FAPAS. It's a UK-based accredited company to check, okay, if the method is really proficient. Okay, so what they did was to send us samples. We checked using our developed method, and then we give them the results. Okay, and then they will check, of course, if the result will coincide with the result that we got. Okay, so we were able to get a satisfactory rating for being proficient in all of these matrices. Okay, so again, just to emphasize, this is actually a DA Biotech project. So it was in 2017 that we were able to optimize the method, okay, by the lab. My lab basically is FEREL, or Pathogen Host Environment Interactions Research Laboratory, and it was funded by DA Biotech. Okay, so it only takes 3 days to complete. And as I said earlier, we were able to extract DNA and then process the sample in just 3 days. Okay, now if we have the DNA, we can do a lot from the DNA molecule, right? Okay, so if we have access now to DNA, we are sure that we also have access to a plethora of data, such as resistance genes, virulence genes, even genetic determinants of transmission, and genotypic serotyping. Okay, so this is the company that I mentioned earlier. It's called FAPAS. It's a third party accredited proficiency testing company provider based in the UK. And as I mentioned earlier, we were able to check Salmonella enterica coming from different matrices, beef, chicken, feeds, and also swabs. And we were able to get a satisfactory rating based on the proficiency test made by these organizations. So yeah, it was in February to March of 2018. Okay. So part, of course, of the DA Biotech project, okay, an expected output for the project would be for us to capacitate individuals, especially coming from the monitoring agencies that, like the National Meat Inspection Service. Okay, so we had a training workshop, and we actually invited personnel from NMIS, also from other agencies. And the goal there is, of course, to build and strengthen the capacity of the monitoring agency for this purpose. But of course, it's still a challenge. We really hope that monitoring agencies like NMIS would adopt the technology for them to have, of course, a faster way of checking on the contamination of meat products. Okay, so of course, not only NMIS, we also invited a Bureau of Animal Industry during that workshop. Sorry. Okay. Aside from that workshop that we had for PCR in 2022, a lamp-based a lamp-based salmonella detection method was also developed by the lab. Okay, this is of course through the funding of the DA Biotech as well, and afterwards, of course, we would like to capacitate individuals once again. And again, we invited personnel from the NMIS and also from BAI. And we taught them the developed method on LAMP. Of course, we know that LAMP, or the loop-mediated isothermal amplification assay, this is a molecular technique that allows, of course, high amplification of target genes within an organism's DNA. And it works differently from PCR. Well, obviously, they both amplify the gene fragment, but the difference is that on the enzyme that are used for the procedure. So for PCR, we normally use a Taq polymerase. It's a thermophilic organism, a Thermus aquaticus, and you extract the polymerase from that organism. That's Taq. Okay, but on the other hand, your polymerase for LAMP assay is coming from another organism. It's Bacillus styrothermophilus, so it's BST polymerase, and the annealing temperature to be used for BST is not as high as that of the Taq. Okay, so you have a stable polymerase for BST, which is much lower than the Taq. Okay, and we found when we developed this assay, we found that this is also sensitive, of course. It does not require expensive equipment, while similarly utilizing, of course, the same genetic marker, INVA-gene, to check on the presence of the organism. So we were able to develop that method, and just last year, I think we were able to hold this capacity building workshop to help NMIS and also BAI to learn the developed method as well. So now that we have the DNA, as I said earlier, we can do a lot of things from the DNA. So our lab also ventured into the use of next generation sequencing, or NGS. Okay, I'm sure many of you are familiar with the technology. And when we say NGS, okay, we can massively sequence a lot, the whole genomes of organisms. Okay, you're able to sequence genomes. It's high throughput, okay, scalable. It, of course, it's fast. It generates large amounts of data for different applications, some of which are, of course, gene mining of genetic determinants for resistance. You can check on the virulence. You can even understand the transmission dynamics of antimicrobial resistance, for example. And then obviously, okay, instead of doing the tedious serotyping or serological assay, you can deduce, of course, the genotypic serotypes of your Salmonella enterica from your NGS data. Okay. So with that, our lab as well, Ferrell, has generated data for applying the NGS, and we were able to get whole genome sequences of Salmonella enterica isolated from farms, from slaughterhouses, from markets that have basically been used traditionally for genotypic serotyping, but we were able to get the data and generate them in a much faster way, and we were able to publish the data just this year. Okay, we targeted Metropolitan Manila abattoirs and markets, And we were able to get insights on the circulating virulence and antimicrobial resistance genotypes. Okay, so just to show you that just very recently, DA Biotech gave us again the funds to do this. And we were able to conclude the project focusing on the utility of NGS to trace antimicrobial resistance, transmissibility, and even virulence of Salmonella across poultry and swine food chains from Metro Manila. So we were able to generate, of course, a lot of data, but the good thing here is that bioinformatics can actually be employed. And yeah, we hope to equip our lab also for this technology, bioinformatics. We have already started. And one of the data generated is our phylogenetic trees that would, of course, check on the evolutionary relationships of the different isolates that we got. Okay, just to give you another example of the data that we have generated from the NGS or whole genome sequencing. This is an example of a single linkage cluster reflecting single nucleotide polymorphisms or SNPs, okay, within serovars, and showing you, of course, the clustering of different isolates, okay, coming from different districts here in Metro Manila. Okay, we got samples from Quezon City, from Caloocan, from Valenzuela, and we check on the different serovars like recent serovar, London serovar, Anatum, and then Infantis. And we were able to check on the clustering of these isolates and serovars within the markets and the slaughterhouses, okay, that we were able to collect our samples. So really, we are able to generate, okay, a lot of data coming from the NGS. Okay. So question is, where are we now? Okay, where are we currently? We're currently, of course, we know that the national service laboratories are still employing culture-based laboratory-based methods in Salmonella enterica detection. But what have we started so far in our lab? Well, of course, Ferrell, our lab has developed a protocol for Salmonella enterica detection that is DNA-based. It only takes a maximum of 3 days to complete. The efficacy of the protocol has been validated by a third-party international proficiency testing lab that I've mentioned earlier, the FAPAS, and that developed protocol is, of course, proficient in Salmonella enterica detection, okay, in different matrices, beef, chicken, swabs, and animal feeds, okay. Now, since the protocol is, of course, DNA-based, as I said earlier, we are able to further characterize the isolates, okay. We now have a database of chromosome and plasmid encoded virulence genes, also the antibiotic resistance genes. It has began applying, of course, sequencing in different studies that we have, and we are now even drafting a proposal for another potential funding by a funding agency. So we hope that we will be able to, of course, accomplish, okay, and develop all of these methods in our lab. But of course, we would like to capacitate or help our monitoring agencies to adopt the technologies that we have developed so far. Okay. So what are our goals? Okay, we of course would like to have a widespread use of the technology that we have developed, and as much as possible, if we can also employ next generation sequencing. Once you have the DNA, then you'll be able to do in situ testing. You can have parallel national guidelines and also standards. Okay, I'm so hopeful with this part of the slide. Okay, if we can have traceability of the contamination, and hopefully we can have this stamp as well on the eggs being commercialized, of course, in markets. If we can have that salmonella-free stamp on the egg shells, then that would be a very good goal, of course. Okay, and yeah, and hopefully we would like to have a lateral flow-based detection method, which would be faster and can be applied in different matrices. So with that, Thank you very much once again. And I would like to thank the funding agency, first and foremost, DA Biotech, for helping us. I think so far from probably 2012, we have received so far 6 grants from the DA Biotech, and we hope that DA Biotech will still grant us a few more millions to finish our goals in the lab. Okay, and thank you, ISAAA, for again the invitation. Thank you very much, everyone.
So thank you, Academician Wendell Rivera, for sharing about the various technologies for salmonella detection. So for our last presentation, Dr. Pantua will discuss the technology development and commercialization pathway. Dr. Homer Pantua is a veterinarian and scientist who has led both antiviral and antibacterial drug discovery efforts. He was a senior principal scientific researcher at Genentech, a pioneer in the development of therapeutics from recombinant DNA technology. Dr. Homer obtained his PhD in biomedical sciences from the University of Massachusetts Medical School and developed a patented technology on virus-like particles as a vaccine platform for human and animal pathogens. He received his DVM from the University of the Philippines. He initially received infectious disease training at the Research Institute for Tropical Medicine. He is currently the CEO of BioAssets Corporation, a pioneering life science company in the Philippines, engaged in veterinary research and diagnostics. He is leading the company's research and development efforts. He's also a project leader of a DOST-supported project under the Business Innovation for Science and Technology, which aims to address the unmet needs in the animal industry. He is a balik or a returning scientist of DOST and is currently involved as a technical advisor in the establishment of the proposed Virology and Vaccine Institute of the Philippines. He serves as a driver and catalyst in establishing research collaborations between academia, government, and industry projects on pandemic preparedness, disease surveillance, and vaccine research. Dr. Homer is very passionate about developing transformative therapies and vaccines for infectious diseases in humans and animals. Let's all welcome Dr. Homer Pantua.
Thank you very much, ISA, for having me here. I also like to thank the Institute of Biology and thank you very much, DA Biotech, for having me here. I'd like to recognize the presence of Director Paul Limson. Maraming salamat po. And it's nice to be here again, UP Diliman. Today, usually I give technical talks, but this time I'll be talking about commercialization pathways, development and commercialization pathways. And it's very nice that given Dr. Vital and Dr. Wendell Rivera has given me a very good background of basically what's the why and what's the how already. And now I'm going to continue on how startups or how companies can actually pursue development and commercialization efforts of this technology. So for today, I'm going to be talking about, I'm going to define what is technology commercialization, what's the commercialization process and the pathways, and what are the essentials for commercialization success. And I'm going to give you a few examples, one in drug development and one in diagnostic kit development. So just to start with, I just want to emphasize to you that technology development and commercialization brings innovation from concept or from idea to the market or market where the deployment is. So what is technology commercialization? So the idea of this talk is basically to stimulate discussion in our later forum. So it's not long. So, but basically I'm going to give you theories and my personal experience in technology commercialization based on drug development. So technology commercialization is the process of transitioning technologies or innovation from the research lab or from the ideas to the marketplace or to the end users. And how does it happen? So for example, it happens from the idea or the concept generation, which is basically why. So why do I say why? Because why is basically when we start with a drug development program or a diagnostic kit development program, the first question we ask is why? Is there a problem? And then if there's a problem, if there's an unmet need, what is the solution? And from the idea, I would like you to I would like us to drive I would like you to drive the attention to deployment. So deployment is basically the success. Deployment is basically the outcome. So whenever we do drug development or technology commercial development and commercialization, we always think about visualizing the success. Where do we want to go? What's our destination? From then on, from the idea to the destination, we move back. How do we do it? Okay, so first we visualize the success. Where do we want to go? And from then, from our idea, we think of a solution. We screen the solution. Is it feasible to do it? Is it something that would directly address the problem? And if it's a yes, we go to concept development. That's the how. So that's what Dr. Windel has done. They have generated a faster PCR platform that could address the challenges of longer or delayed diagnostics. So the idea generation is from what Dr. Vital has presented to us is basically why there are challenges in food safety. For example, diagnostics. So we basically answered those questions already. So concept development, we make a proof of concept. Now I think Dr. Windel's technology is beyond the proof of concept already. They've done the proficiency testing, so they're a little bit farther in the development pathway. So from the concept development, we make a product development. So now I think the next for Dr. Windel's group and or DA Biotech is to develop a product, to make a prototype wherein we can actually proceed to the market. So, but there are cycles we need to consider during product development. So product development involves validation, assessment, and optimization. So during this phase, we keep on testing the product, not only the efficacy, but also can we actually produce it? So one of the things why drug development or diagnostic development failed is because we cannot scale up, we cannot produce, we cannot establish a process. So those are the challenges. So we don't only have to look at the effectivity of the product, but also can we actually mass produce it? So we go around, validate, assess, and optimize, and ask again until we make the best product. And from then on, we go to deployment. And deployment involves a lot of things, and I will discuss that later. So this is just the technology transfer cycle. This is basically used by many innovation and technology development department. So it starts with the research and development. That's where ideas happen. And then we make an invention. And then we evaluate that invention and then ask whether are there proprietary strategies for that invention? Because sometimes a lot of companies would always consider IP protection. So from then on, from the idea, we can actually market the idea. We don't have to get to the product to market it. The idea itself is already marketable. There's a proprietary strategy or a bargaining power that we can take advantage from just the idea itself. From then on, we can find licensing opportunities, and I will discuss that later. And then the product development through licensing and use and economic growth. So what are the factors influencing commercializing strategy? So there are three that we always consider. One is resource, which basically looks at companies or capacities that are in synergies, or the capabilities and capacities of those institutions. And financial resources, one of the things that we consider. Can we actually fund this development program? The next is intellectual property. So a lot of companies always think, so is this technology novel? Is there an distinct element that would protect this idea? Because if there is a distinct element that would protect this idea, and it answers a why, we can use that as a bargaining power to make that idea a business already. So next is the transactional cost. And transactional cost is basically complementary assets. And these are the capacities, production capacities, research and development capacities. And I will discuss more complementary assets in detail in the next slide. So what are complementary assets for commercialization? One is product development. Product development involves the research and the process development and production. Process development. So one of the things that hinders startup companies and academias from moving into development is the cost or the capacities. Other than the cost, we have to determine whether do we have the capacity and capabilities to answer questions, to look at hypotheses, so something like that, because it involves a lot of skills, personnel, and technology. So that's very expensive too. Another is production. It involves process development and the actual manufacturing itself. So it's very hard for a small company or for academia to build its production facility. That's why we have to venture into other options, which I will discuss in the next slide. And sales and marketing. So you have you did the research and development. You can actually address production, but sales and marketing is different because building a sales force is also very expensive. And it also, you have to make sure that you can cover all the market opportunities. So those are the complementary assets. That is very challenging for an academia and a small startup company to establish. So what are the ways to build complementary assets? One is vertical integration or outsourcing. So vertical integration is basically if we don't have the research and development capacities, we're going to build it, but it's very expensive. The production capacity, we're going to build it. But for example, in a vaccine development program, it takes millions of dollars to develop a production facility. So, but the advantage of having a vertical integration or outsourcing means we're going to use atoll manufacturing to make our vaccines or drugs or kits. But what's the advantage for this is we can keep control of technology rights and returns. So basically we own the technology and we own all the revenues. So that's one of the things, but it also takes a lot of cost or investment for the startup company. So the disadvantage is that it involves high investment costs and needs knowledge of complementary assets. Not only you don't need only to have the investment, the availability of investment, but you need to have the skills, and it's hard to get personals to, for example, in a vaccine development program, in a fermentation scientist, process scientist, QC scientists. So all those you have to think about. That's why it takes a lot of effort and investment to do vertical integration or outsourcing. But outsourcing is a little bit cheaper than vertical integration. So one is cooperation. So for example, if an academia has an idea, what we can do is we cooperate with a startup company, or you can spin up to build a cooperation or partnership between institutions or entities that have the complementary assets for research and development and production, and also the one that can give you a sales and marketing force. So what's good about this is that you can do this through licensing, equity-based partnership, or joint ventures. What happens with this one is that you have a technology, you find a partner, and you out-license or in-license those technology to a partner. Or these partners will put an investment as an equity to your technology. Or the easiest way is when you have an idea, like Dr. Windel have an idea, you can sell the technology right away. So what as a startup company, I can actually deal with the university
through licensing, through a cooperation, or you can directly sell the technology to a startup company. So what are the licensing strategies? So these are based on experience. So one is option to license. Is basically it grants the exclusive rights for a limited time frame. And it allows the partners a chance to evaluate the availability of opportunity. So in this scenario, what happens is, for example, a vaccine platform or a diagnostic platform, it's you send it for an options to license for a period of 5 years, and for that 5 years, when that ends, it gets open to another entity. And then, but the advantage of that for a startup company or for a partner is that we are able to evaluate the technology, whether, for example, a vaccine platform, whether it will work for several systems or for the diagnostics, for us to be able to determine its effectivity, reliability, and commercial viability. So it gives us time to evaluate the technology. The exclusive license basically grants the exclusive rights, everything, most often to a patented technology in exchange for some financial consideration. So what happens is that, for example, if you have a vaccine platform technology, you license it exclusively. Usually, a licensing agreement costs at least $1 million for a technology that works already. So terms often include the royalty-based sales. So after that licensing agreement, so they do royalties and payments on a milestone basis. So as the technology progress, as I've told you, the idea itself is a business already. So once from every milestone, you give payments to the technology provider. So non-exclusive license is basically it allows non-exclusive access to the technology by numerous industry partners, sabay-sabay kayo. And then terms are typically include a royalty based on sales, which are lower relative to exclusive because there are many entities that are involved. So, but this one, it do not often include due diligence provisions, but may include an agreement maintenance fee structure. Compared to exclusive license, it needs some diligence provision for future years, such as milestone events and payments, as I've said before. So what are the phases of technology development and commercialization? So it's divided into development and commercialization. So when I came here, what people think is that research and development are the same thing, basically. But research is basically the discovery phase, and development is basically developing the production, developing the process, and moving it into regulatory and commercialization. So that's how we do it when I was still working in the biopharma. So what does technology development involves? So it assess and evaluate need and market analysis. From the very start, what we do is we have a competitive intelligence group. So basically, these are private eye what their job is to go around conferences, conventions, and look at technologies and look at the landscape. For example, if we're looking at developing antibiotics or diagnostic kits, they're looking at what's available out there, what's the landscape, what's the commercial viability, and what's the need. For example, the why, what are the challenges? So we need to establish that. The next is the research and conceptualization. So basically, this is our early stage research. We ask a question, we look at the biology. If the biology is right, then we proceed to doing a proof of concept or the prototype. And then the development. The development involves, for example, in a drug, it involves preclinical studies. It involves efficacy, safety in preclinical animals, and then scaling out. So for example, in a drug development or in a vaccine development, can we actually produce it? Because one of the things that we face in drug development, we can actually make it in small batches, but there are challenges in making huge batches to serve the market need. So those are the things that we do in the technology development phase. Now we go to the commercialization. Even before the commercialization, we establish intellectual property protection. So at this stage, even in the development and scaling, we start the patent. And in Philippines, is it first to file? It's first to file, right? So what's important about intellectual property protection, it fosters trust between entities and the technology provider. So next is productization. Productization means we're building like a prototype of the product. And then distribution and marketing. So this goes to the sales force already, the sales and support, and continuous improvement. So the technology development and commercialization does not end to the sales. We have to innovate. We have to do the next generation, or we need to make, for example, in Apple, they actually make the product that will kill the previous product. So we need to continuously improve. So what are the considerations in technology development and commercialization? So I'll just expound the pathway that I've showed you. First is the assessment phase. So what we do in an assessment phase is we look at value proposition. So basically looking at, is there an unmet need? Are there distinct elements in the technology that we are going to use? So but there are distinct elements, we have a competitive advantage. So if there is competitive advantage, is there a proprietary strategy? So can we actually protect the technology? Is there a market advantage on this that will support competitive advantage? Next is market opportunity, which basically looks at clear market fit. So for example, in this PCR based detection platform that we have from Ferrell, is there a clear market fit? And I think it does. It does, because what the problem with Salmonella detection is that it takes a long time to get to the yes and no. And what's good about this technology right now is that it does not stop to the yes and no. A lot of technologies will prevent you to the next phase, but this one still allows you to do genomics. It keeps the sample, the integrity of the sample in such a way you can still generate more information that would give meaningful information to understand Salmonella in the Philippines or wherever. Is there sufficient market? So this is commercial viability. So if we do that, are there going to be a lot of adapters, a lot of end users? Basically, for startup companies and biotech companies, this is the dollar or peso sign, right? So the implementation phase involves business model. So how are we going to move it into the market? How are we going to get more funds to produce the technology? Proprietary protection, again, IP asset definition. We have to differentiate from the prior art, and then the license revenue, partnerships, and follow on milestones. So as I've told you, the technology itself without the product is a business already. So in the US, when a lot of companies just keep on making ideas or concepts into proof of concept, and then they out-license that to large companies with the hope that these large companies can bring it to commercial landscape. So this is an example of a drug development and commercialization. So what the technology research for startups, for example, in the Western world is our academic and medical centers and federal laboratories. And I think in the Philippines too, academic and research institutions are the technology resource, and national laboratories are should be technology resource. So the first phase is discovery and preclinical stage, which is basically, in these terms, we identify the unmet need. The next phase is to identify the solution, which is basically the how. And then it's also under the discovery and preclinical stage, we do all the discovery biology. For example, the target identification, we identify tool drug, the companion diagnostics, the proof of concept, the screening. At this stage, we also do IP protection already. We improve the efficacy, safety, and toxicity, and the process development. So at this stage, up to preclinical. And then in clinical stage, we do the phase 1, 2, 3, safety and toxicity, efficacy in small cohort, and then in large cohort. And once we have the yes of FDA, we go to the regulatory, and once we have the yes of FDA, we go to production and sales. So where I came from, when we do FDA applications, right when we are talking to the FDA, the first batch is ready already. So once the FDA say yes, it's going to go to the patients right away, the next day or week. So that's how fast it is. And one of the examples here that we've done in my previous work, we've done therapeutic antibodies, but I'd like to cite a gram-negative antibacterial drug development program where we identify the unmet need, which is basically gram-negative infections. For example, Pseudomonas, Acinetobacter, and a lot of Enterobacteriaceae infections. And the idea is to identify solutions, antibiotic, but not just antibiotics. We want to find a novel, an antibiotic that has a novel mechanism of action. The idea is because for the past 50 years, we have not identified a novel antibiotic or something that has a distinct mechanism of action. Most of the antibiotics that we have right now are basically targeting, are just second generation, third generations of some sort. But we have not found something that is novel for the past 50 years. So what we do, we have a target, we identify a target, we understand the biology. So where we came from, we are not a generic company. So we basically start with the science, we identify, we understand the biology and genetics of the bacteria. For example, the first question that we ask is essentiality. So essentiality means, is this gene essential for the survival of the bacteria? If it says yes, through knockout experiments, then we proceed to target development and identifying a tool compound or a tool antibiotic that will give us an idea on how to target this gene. So then if we have that, we have a new molecular entity that could be in-house developed or it could be licensed. So I told you that we identified the target already. So we can develop our own, or we can actually shop around the industries and look for something that will target this gene. So it could either be in-house or licensed. So what's the difference between in-house? In-house, you invested a lot of money in R&D. But if you license, someone else actually invested in the more expensive phase of the development, and now you just pay them with licensing fees. So mas mabilis. So it's going to be faster if it works. That's why there are due diligence, validations, and assessment. Then if it's good, we go to clinical, regulatory, and commercialization. It's basically the same. So now in diagnostic development, we're gonna now that, because we are talking about Salmonella, what can we do with the technology that we have that IB Biotek supported? So we identify the unmet need. So there are challenges in Salmonella detection. There are challenges and problems in bacterial infections. For example, salmonellosis has been, it's like around 93 million every year in the Philippines. I think based on the data from DOH, there was around 42 to 46% increase from 2022 to 2023. And one of the things that we should do is actually surveillance or detection to protect our communities, and also to protect the livestock, which is basically important for food security. And one of the things that we need is diagnostics. And not just diagnostic, but a rapid diagnostics that would make 7 days to 3 days. So that answers the unmet need. So what they did right now is they did a design, a proof of concept. Now we need to make a prototype. Okay, if there is something, and I think we should invite startups, we should invite private entities to work with UP and to work with IB Biotek, and how we can actually move this into commercialization. So that's where we are right now. Because with the proficiency, satisfactory so proficiency testing, I think we are very close. We are very close to moving it. And I hope the Biotech will keep on helping the the academia. So we're very close. So now what I'm gonna tell you in the last few slides is that I'd like to take the opportunity of introducing you Bioassets Corporation. It's a pioneering life science company. It's a startup company. It's about 9 years old already. Before, they know us as a diagnostic company only, but basically, we're not just your ordinary diagnostic company. We have R&D programs that are geared towards answering problems of the animal industry, and we're also going into humans also. But basically, Bioassets is like a contract research organization with the vision of having an R&D programs that addresses infectious diseases here in our country. So how do we approach innovation? And the way we approach is we follow the triple helix model of innovation, where government, academia, and industry collaborates. So when industry and academia collaborates, for example, industry such as us and academia such as the university and other research institution, we develop technology transfer and we foster innovation. And when academia and government works together, we put a legal framework for funding and for innovation. And when government and industry works together, we establish a policy enabling environment. When all of three works together, we support socioeconomic growth. And that's how we approach innovation in our company. So what are the essentials in driving innovation? So what we do is we access government support. We look at research funding. We establish research centers. We consider advisory boards, because advisory boards are very important in introducing industry perspective into research. And academia industry joint projects, such as we have right now with UP Diliman and UP Los Baños. We have internship training programs and knowledge exchange platforms, IP protection tech and tech transfer, which is important in fostering trust between entities. And of course, this won't work if we don't have continuous engagement to develop technology development, to establish technology development and commercialization in our country. And what are the essentials for a successful commercialization? So basically, I always advocate for collaboration. And what I always say is that collaboration between various stakeholders, we need this collaboration to support development. And it involves researchers, biologists, engineers, marketers, investors, regulatory bodies, and these regulatory requirements and compliance standards to ensure safety and legality of the technology. We just don't do or execute producing a technology or a product. We have to make sure that we ensure safety and legality. And with that, I always want to encourage everyone to collaborate. I always believe that encouraging collaboration and fostering innovation is important in technology development and commercialization. In our company, we don't have the best ideas all the time. So what we do is we create an environment where great things can happen. That's why we always invite everyone, let's collaborate for a better industry, a better Philippines, for you, our families, and myself. Thank you.
So thank you, Dr. Pantua, for your educational presentation. We would like to call Dr. Vital and academician Wendell Rivera for the Q&A. Please come here to the stage. And for our on-site and online participants, please prepare your questions to our research speakers. Thank you. Our Q&A will be moderated by Dr. Ivan Christian Imperial from the UP Diliman Institute of Biology.
Okay, so good morning, everyone. So first of all, we'd like to thank again our speakers for giving us very informative talks and well integrated lectures. So as mentioned by Dr. Pantua, we started with the whys, so the challenges in food safety, the potential problems with the talk of Dr. Vital, and this was followed by the how or the proposed solutions as discussed by Dr. Rivera. And then we ended with the bigger picture in how to further develop technology and commercialization as discussed by Dr. Pantua. Okay. So we can now start our panel discussion. So for those who are here at the institute, we encourage you to just approach the microphone if there are any questions. And then from the Zoom, kindly type your questions. So we now have a lot in via Zoom. Okay, so from the Zoom, from an anonymous attendee, thank you for the excellent presentation. I've learned a lot. However, I know that in the early 2000s, UP Los Baños already developed and patented detection kits for foodborne pathogens. I'm just curious, how is the different from the commercially available and patented PCR-based detection kits by Biotech UPLB? Thank you.
Unfortunately, I'm not so familiar with the method produced by DA Biotech. But well, as emphasized in my presentation earlier, the technology that we developed would really go far in terms of not only detecting the pathogen, but we can at the same time do serotyping, and then check on the virulence genes, even the antibiotic resistance genes, to check on antimicrobial resistance dynamics, transmission dynamics. Sorry, I'm not familiar with the UPLB Biotech work on the detection kit.
Okay, so thank you very much. So another question, given the various opportunities for food contamination, particularly with Salmonella occurring at different stages from pre-harvest to post-processing, how do you plan to integrate the Salmonella detection kit into our food system? Will there be a time that the kit will also be accessible to our farmers or even our consumers, or will it only be available through testing facilities?
Yeah, that's a good goal. Obviously, for a consumer to have access to the detection kit. Okay, but that would be really far, I think, at this time. But at least, as I mentioned, the monitoring agencies like NMIS or BAI for the poultry industry should have access to this detection method. In the last slide that I flashed, I also showed a lateral flow detection platform that can actually be used. So if we have that kind of platform, then obviously our consumers can also have access to that kind of detection kit.
Yes,
If I may add, I think the question is when to use the kit. I think it is ideal to use the kit at each point, like from pre to harvest to post-harvest. However, we have to look at the cost, of course, the practicality. So it would be best to use the kit at post-harvest or even before the consumers will consume it. Thank you.
So thank you very much. So also, so as a follow-up to that question, so I think this for Dr. Pantua. So given that the benefits were mentioned regarding the application of potential solutions at different points in food production, so how or what will be the challenge to make the technology more accessible in our setting, especially that we're not considered as affluent as a country?
Yeah, I think the technology is there already. The proof of concept is convincing already. What I think is that developing this as a commercial product is not that hard. It's a PCR. When we see that the misconception that we always have is that when it's a PCR, it's a polymerase chain reaction. Oh, it's expensive, but it's not. It's not. So there are many ways right now how to produce the master mix, how to produce all the reagents and ingredients that we need for this. What we need right now is a technology adapter. So technology adapter that believes in the platform and believes that it can be taken commercially and it will be used by the end user. And I think it's doable. It's doable, especially that we develop it here. So we don't need to make all the reagents here. Now that it's easier to import stuff and everything, if we could make all the reagents, that would be great. But we don't have to. We don't have to, because one of the challenges in making all the reagents is that can we make the polymerase cheaper? If we cannot, it's gonna make our kit more expensive. So there are balances between importing the reagents and others. So for me, I can be in our group we are also a technology adapter. The way I look at this is basically we adapt the technology. We make the master mix and put a prototype, and then we do a trial with different. For example, I think when you were still at DA, Doc Limson, you support ring trials. So we asked different entities to test the product and technology and ask whether can we actually have the same result like you did with papas. Now using the prototype, and when we see the same satisfactory result, I think we're there already. We go to the FDA, and we go to the FDA while doing the ring trial. I think it's not expensive to do it. We just need a believer who will adopt the technology.
Thank you so much.
I'm a believer.
Thank you so much. So anyone from the audience, from the live audience? Kindly approach your microphone, please.
So, hello po, good afternoon. This is a very beginner question, I mean, on my end. I think this is a question for Dr. Pantua. With your expertise in drug development, so I've been asking this myself a lot, especially Mati's proposal, laging drug development, finding antibiotics in the environment. So in developing drugs, especially antibiotics, the trend is to find something that can potentially inhibit or kill the bacteria, but screening novel compounds takes a lot of time and limitation. So my question is with especially sa current development ng technologies natin is what's stopping us from making the compound itself instead of finding it in the environment such as plant or fungal extract? Yun nga, especially sa technology natin, parang what's stopping us from synthesizing the compound instead of finding it?
Good question. Very interesting. So we work with I worked with natural products before in the US, and one of the things that I noticed here in the Philippines is we look at soil. We look at soil. We look at the environment. If you look at soil, based on my limited knowledge, you're going to find the same thing over and over again. What we need right now is an antibiotic which has a distinct mechanism of action. If you find streptomyces, if you find other fungi that produces antibacterial, it's going to be the same. What we need right now to address AMR is something different. So it's hard to find that, and we need to consider other sources than the soil. So I think marine is a good source. I think MSI has a wealth of extracts that we can actually pursue. I think the DA Biotech should pursue that. It's a wealth of extracts. It's different from the soil. So we just need to make a very intelligent assessment of what you have to make sure what you will get is will actually address the problems that we have right now with the escape or the other most especially the gram negatives. So one of the things that we always we have to consider also is Yeah, you find something, can you improve it? In the Philippines right now, admittedly, we don't have medicinal chemists. Medicinal chemists are very important in doing structure activity relationship studies. Now you have an extract, you do a mass spec, you do a lot of characterization, you find your molecule. What are you going to do next? You have, that's not an antibiotic yet. That's a new molecular entity that has antibacterial properties. That's just a tool. It's not the product yet. You have to make sure that your efficacy is within reasonable range compared to the toxicity. Because if they are very close, then it's not an antibiotic yet. It's just a molecular entity that has some antibacterial properties. That's why we need to have a very, very good medicinal chemistry to work on SARs to widen the difference between the efficacy and the toxicity of the molecular entity. And right now, I think that's one of the things that we are looking at. I was here in UP last week. We're talking about, do we have medicinal chemists? Yeah, you have this. You have, oh, nice microcycles with some lipid tails. Oh, that lipid tail is very long. It's going to be toxic. I bet you. I bet. That's why we need medicinal chemists. And if you're telling me that your MIC are double digits, drug antibiotic development companies will say, forget it. You have to target single digits to like micromolar amounts to make sure that you can actually bring it to the clinics. Okay.
So much. So another question. Anyone from the audience? So let's move again to the remote questions. For Dr. Vital, given the discussion of the role of the government in food safety, so I think the question is, what are the potential roles in addition that we already have right now that the government can actually go into? So what more can government do to help in terms of addressing food safety?
I think, as I mentioned, we already have all those guidelines present, and the government and especially the DA and the DOH are making new national standards. So I think another thing or most important thing that we can do or the government can do is consumer awareness, or to translate these guidelines or national standards in a way that all of us, even not scientists or not researchers, can understand what these guidelines are, and that we would know that we are protected by these guidelines. As I have mentioned earlier, that we all have a role in keeping foods. So I think very important for us to be able to know all these guidelines that we have. So consumer awareness.
Thank you so much. Another question, can the technology discussed, I think for Dr. Rivera, I think, can the technology discussed also be applied in virus and viroids in plants?
You mean the DNA technology,
I
right?
think,
Yeah,
so.
of course. So if you have, well, it depends on the virus. Obviously, we have DNA or even RNA virus. So if it's a DNA virus, then you can easily extract the DNA. If it's an RNA, you have to convert it to a DNA molecule, and then you can do the usual PCR. So yes, DNA technology, especially PCR-based technology, can be used for viruses as well.
Okay, so just a reminder for those who are sending the questions. So I'm kind of screening this impromptu. So some are very similar questions. So another question, what method or validation guidelines or standards were followed during the development of the technology?
That's for me, right?
I
Okay.
think so.
So yeah, I mentioned in my presentation earlier about the proficiency testing. I forgot to mention about parallel testing as well. So in parallel testing, what we did was to collaborate with the National Meat Inspection Service. So they gave us samples. We checked, of course, for the presence of Salmonella in those samples, and then we gave our results to NMIS to cross-check if their positives are also positives from us. And yeah, we got a high rate of positivity from the samples that we received from the NMIS. In fact, of course, the usual, we got higher positives for the PCR than the culture-based method done by the NMIS. And that's, of course, self-explanatory. Okay, so that's the value of using molecular method to detect your pathogens. And at the same time, the FABPAS that I mentioned, we had this proficiency testing with the UK accredited third-party company. They also sent us different matrices, okay, that included beef, other meats. I'm not sure if that included swabs, I think. And then we checked for the presence of Salmonella. And that was cross-checked by their company. And then we got a satisfactory rating for the validation of the technology.
Thank you so much, sir. Sir, there's also another follow-up. Is this some costing or ROI study that was done? So I think that goes first.
Yeah,
Dr.
that's why I'm collaborating with Homer. Homer.
The cost. So right now it's I don't know the, but I think it's very it's reasonable. It's gonna be reasonable. I know because in research we always do research tools, and I think if you compare this with commercially available kits, it's gonna be I think the cost that we will that we will need to produce this will be reasonable in such a way that our NMIS, our BAI will be able to adopt the technology. It's going to be competitive, I think. I don't know the exact, but we can make it really reasonable that we won't need to think about, oh, how much is the imported ones versus the same, versus ours that has the same performance. It's going to be reasonable.
Thank you so much. We still have a lot of questions, but I think we have time for a few more. So this one is in terms of the antimicrobial resistance that was mentioned. Can with all the technologies that we have, is there some cost-efficient method that can be suggested to laypersons in terms of addressing antimicrobial resistance? So,
I think the most cost efficient, there are many factors affecting AMR. It's misuse and disuse of the antibiotics. One of the things that would help humans and animal health alike is policies. Policies will help prevent will help the AMR reduction program, and awareness of our all stakeholders and communities that in humans we should avoid misuse of antibiotics. In the animal health also, as you have known, a lot of in the industry, not just the Philippines, all over the world, there's a lot of use of antibiotic in feeds. So, but right now, if we can have a policy that prevents that, actually, I was in the PBMA, Doctor. I was encouraging people to not use colistin. We should save colistin for humans because colistin is the last resort, last treatment of resort. And if everyone knows what happens to a human that is treated with colistin, it's highly nephrotoxic. And if that's the last resort, and there's AMR already, it's gonna be it's really detrimental to the person who will use it. That's why policies are very important. I think we have now regulations that look at vet drums, yes, something like that. So policies are very important. That's the most cost effective, I think. But it's not just the solution. but it could start from policies to prevent the spread of AMR and help the reduction program.
Just in relation to that, I just would like to share that kasi Dr. Homer mentioned policies. So the APAFS is currently developing antimicrobial resistance risk management control options, Philippine national standards for this year. So we are just starting, and I hope we could develop it for this year.
Thank you so much. So, any other questions from the live audience? Para hindi lahat yung baka meron pa gusto magtanong. Yes.
May question na ako kay Academician Rivera. Siyempre po yung ating objective in developing these technologies to commercialize it. Since ang Institute of Biology is a technology developer, does UP Diliman have a special unit that will handle the marketing once the technology is already mature?
I think for the IP issues, intellectual property, we have an office. We call that Technology Transfer Business Development Office, or the TTBDO. And at the same time, that office actually helps scientists as well to be linked to companies for commercialization. That's the only idea that I have. This is through the Office of the Vice Chancellor for Research and Development. Maybe Homer, with your discussion with the university, probably have a better idea.
I think they have the CTTE, or the Center for Technology Transfer and Entrepreneurship. So right now we've been dealing with them. We've been working with CTTE in UPLB and hopefully in Diliman also. So as a startup, we are open to technology licensing. So we are not just an R&D company, we are an adapter. So if with the University of the Philippines, we deal with CTTE.
Okay, so any other questions from the live audience? I think we have still time for just one last question. So let's go again. Sure.
Just to address the antibiotic development, I'm not saying we cannot do it. We just need, I think this is an opportunity for the university to develop medicinal chemists. Because if we want a really good drug development program, we should develop medicinal chemists.
Okay, so I think thank you very much for that very informative discussion and answering questions. As much as we want to consider all the questions, so we have limited time, and there are some questions that are overlapping. But once again, we appreciate the time and effort devoted by us with our speakers, and thank you very much for our hosts and the sponsors for this talk. Thank you so much.
So while the participants are answering the evaluation, we will now proceed to the closing remarks by the new Director of DABPO, Dr. Paul Limson. So Dr. Paul Limson is currently the new director, as I mentioned. He obtained his Doctor of Veterinary Medicine degree from the University of the Philippines at Los Baños, specializing in large ruminant and lovebird practice. With a career spanning several decades, Dr. Limson has held key positions in various government offices, including his previous position as the director of the Bureau of Animal Industry, where he co-led and introduced policy reforms for the development of the Beef Cattle Roadmap. Known for his expertise and dedication to the welfare of animals, Dr. Limson has played a crucial role in implementing animal health and surveillance programs for economically significant diseases, and has contributed to the Genetic Resource Improvement Program for beef cattle and small ruminants. Dr. Limson is a member of the National Genetic Resources Improvement Program Technical Group and remains an active member of the Philippine Veterinary Medical Association, Philippine Society of Animal Science, and Japan Overseas Cooperation Volunteer Philippine Animal Science Association. He has also served as the national coordinator for the conservation, utilization, and management of domestic animal genetic resources. We are honored to have you today, Dr. Limsan.
Good morning, everyone, both in virtual and physical. I see familiar faces, the Homer. Medyo matagal-tagal na tayong nakakasalubong pala. Ngayon, I think magkakasama for future. So, technically po, I am less than a month assigned sa biotech program office. So it is a high learning curve po dapat sa akin. Pero I think hindi naman po masyadong mahihirapan. So let me read po the prepared speech. To the International Service for the Acquisition of Agri-Biotech Applications, or ISAAA, represented by Mr. Panfilo de Guzman, Dr. Ian Kendrich Fontanilla, Director of the University of the Philippines-Diliman Institute of Biology, distinguished speakers, colleagues from the government sector, young researchers, fellow advocates for cutting-edge scientific research, and to those who have joined us all here, both physical and virtual, a pleasant good morning. I would like to commend ISAAA for providing us a platform for the stakeholders to learn more about the technologies developed by UP Diliman Institute of Biology, the PCR Based Detection Kit for Salmonella on meat, headed by Dr. Windel Rivera. Salmonella contamination, particularly in meat products, poses a serious concern worldwide as it can result in food poisoning and other health issues upon consumption. According to the DOH, cases of Salmonella in the Philippines increased from 9,000 in 2022 to 13,000 in 2023. Conventional methods for detecting Salmonella are first very expensive, as we have proven at the Bureau of Animal Industry previously, time-consuming and very, very labor-intensive, potentially allowing contaminated products to reach consumers before identification. However, the advent of PCR technology provides us with a more powerful tool that can swiftly and accurately pinpoint the presence of Salmonella in meat samples. This particular innovation is indeed a game changer in the field of food safety, enabling faster and more reliable detection of pathogens to prevent the spread of diseases and ensure public health. In the current landscape of biotechnology, where we encounter a multitude of challenges, it is imperative that we collaborate to devise innovative solutions to tackle the pressing issues at hand. I therefore urge all participants, particularly young researchers, to persist in the pursuit of groundbreaking research or researches in biotechnology. Be aware that your efforts have the potential to significantly improve food safety standards, ensure public health, and advance agriculture industry as a whole. Once again, I applaud the efforts of ISAAA and the University of the Philippines-Diliman Institute of Biology for spearheading the seminar. It is events like these that we foster knowledge sharing, and we at the IB Biotech Program Office are committed to supporting everyone's initiative in promoting innovation, collaboration within the scientific community. Maraming salamat po sa pagpapaanyaya at mabuhay ang Pinoy Biotech.
So thank you, Dr. Limsan, for your closing remarks. So for those who haven't completed the evaluation yet, you still have time to finish the form. And to our on-site participants, please stay for the photo op and the lunch. Again, we would like to thank everyone for attending this seminar. We would also like to thank the BPO, the UP Diliman Institute of Biology, and all our speakers. So make sure to follow our Facebook page, fb.com/ISAAA Pinoy Biotek. And also follow ISAAA.org, ISAAA.org. So thank you.